Hello, good afternoon. We're going to wrap up lecture 10, lecture 9 today, and hopefully we'll finish majority of lecture 10 as well. Let's start with a few uh, conceptual questions here to refresh our memory of what happened in our last class. Over here we have a plasmid, and all these description on the outside is telling you where the restriction enzyme recognition sites are. And over here, the question is asking what happens if you treat the plasmid with EcoR1 and BAMH, and then you separate the products with gel electrophoresis, how many bands will you see? So if you are treating it with EcoR1 and BAMH, you'll be having a cut right here and here. And since this is circular DNA, when you cut it twice, you actually get two fragments. So one fragment over here, and then a second fragment over here. And if you run it on a gel, you will see the shorter fragments will migrate further down the gel, whereas the longer fragment will be closer to the well, to the top where you load your sample. So the answer here is two bands. Let's take a look at the next one right here. Um, there are two DNA samples with an STR polymorphism. An STR polymorphism is referring to the short tandem repeats, and they might be repeated different number of times, different individuals. So in sample A, we have a CAG repeated 10 times, and then in sample B, we have a CAG repeated 15 times. So this one is longer. It's actually 3 times 15, which is 45 nucleotides long. And over here, this is CAG repeated 10 times. So 3 times 10 is going to be 30 base pair long. And so again, if you try to separate this using gel electrophoresis, the shorter fragment will be able to move down the gel more. And the correct answer in this case would be sample A, will be further down the gel because it is shorter. Here is another question. We have a DNA ladder, which is what this is. This is the ladder. It's a prefabricated DNA of certain length that you can load into one of the lanes in your gel as a reference to compare your own DNA sample with it. So over here, this one is a DNA sample with two recognition sites, and we try to figure out the original length. So over here, this one is between 8 and 10, which means this is 9 kilobytes. And over here, this one is 6 kilobytes. The thickness doesn't really matter, okay? This is just the location that is what we're interested in. And over here, this is between 2 and 3, so that's 2.5 kilobytes. And if you add them up together, that would be 17.5 kilobyte, kilo base pair. Sorry, I keep on saying byte. Thinking about computers. Kilo base pairs. Okay. So that's how you use the ladder. You can use it as a reference to compare your DNA sample with it. Okay, so this is another kind of polymorphism. Remember, polymorphism is just any kind of DNA difference that can be detected between individuals. So this one is called RFLP, which stands for restriction fragment length polymorphism. So it is a detectable DNA difference in the length of the fragments that you create when you treat it with a restriction enzyme. Right? Here is a very simple example. Over here, we have some DNA from a crime scene, and this person's DNA has two recognition sites, okay? CCGG. So that's a recognition site for some restriction enzymes. Doesn't matter which. So if I take this sample and I treat it with restriction enzyme, okay, treat with restriction enzyme, 1, doesn't matter what it is, then it's expected to cut it twice and you would have three fragments, okay? One fragment, two fragments, three fragments. So you will see three separate bands on the gel. Now, this is someone else's DNA, a suspect's DNA, but this person happens to have, let's say, a neutral mutation. Okay, neutral mutation. And the mutation will have no effects on the proteins, but it will change the recognition site. And so it's no longer CCGG, it's CAGG. So if I treat this sample with the same restriction enzyme, this restriction enzyme will no longer recognize this as a cutting site. It's not CCGG anymore. So it's going to leave that alone, and then it will only cut here. And now you have one fragment and two fragments. Okay. So by comparing the length and the number of fragments that you generate by treating a sample with restriction enzymes, you can distinguish the sample between individuals. Okay. Assuming that they have mutations at the recognition sites. Okay. Now, this is an oversimplification. In real situations, you are not just going to try to treat the sample with one restriction enzymes. Uh, you're going to use multiple restriction enzymes and you're going to digest multiple DNA fragments, um, multiple DNA samples to create a unique banding situations. Okay, don't worry too much about this. The key thing to realize here is there are different kinds of polymorphisms, and depending where you're trying to do DNA fingerprinting, or if you're trying to use other applications, these polymorphisms will be um,
So
uh,
this is another way of detecting any individual, like
exactly.
polymorphism, and this way this.
That's right. Yeah. So you can do STR, analyzing the STR polymorphism, right, by looking at the short tandem repeats, or you can do restriction enzyme length analysis. Okay, it really depends on the situation. Okay, like we're not going to go into details of when we use which, but just be aware of the fact that there are multiple ways of approaching this. We did this last time already. Expect to do something like that on the final test, and maybe on the quiz too. The quiz is on Wednesday. Final test is next Wednesday. Okay, next Wednesday. Okay, we're gonna finish the lecture by talking about gene therapy. So gene therapy basically involves introducing genetic materials into a person's cell, and depending on the target of the cell, depending on the kind of cells you're targeting, rather, it could be somatic gene therapy or germline gene therapy. As the name implies, somatic gene therapy affects bodily cells that is not going to become sperm or eggs, whereas germline therapy is going to affect germ cells, which you learn is going to undergo meiosis to become the sperm and the egg. Somatic gene therapy will only affect the person who's receiving the gene therapy. It will not get passed on to future generations. But germline gene therapy, because it's affecting the sperm and the egg the person produces, it will get passed on to subsequent generations. For that reason, germline gene therapy is considered as unethical at the moment. Okay, for many research institutions, right? Because you don't want to be altering future generations without their consent, right? Like you can't alter the life of a person uh, before the person is born, so to speak, right? And um, and that is not an area of active research, but even though we have the technology to do so. Somatic gene therapy, on the other hand, is okay, ethically speaking. It's gonna affect the person who's receiving treatment only, and the gene therapy has three main goals. Okay, number one It aims to replace a non-functional gene. If you are missing the gene that makes you good hemoglobin, let's put a functional copy in there, then you will start making good hemoglobin. Right? Problem solved. It can be used to suppress an overly active gene. If there is an oncogene that's always telling your cell to divide, then maybe we can suppress the oncogene so that it's not always green light, so to speak, right? And you can slow down the cancer progression. And in some cases, we want to use gene therapy to modify the clinical course of a condition, right? So that it's slow down the progression of a disease, or uh, uh, it's hard to reverse the disease in many cases, but like alter it in a way that it's going to be manageable. It's gonna minimize the effect on the quality of life, right? So we're gonna be looking at these three things in more details, starting with the first one. So gene therapy is at the beginning of it was primarily used as a way to treat diseases where you have a loss of function mutation. Okay. Loss of function means, there is a mutation that causes like a nonsense mutation or missense mutation that causes the protein to no longer function the way it's supposed to. Okay, the protein is created, okay, but it's not doing its job, or only half of it is created because we have a nonsense mutation. Okay, so the logical thing to do here is we deliver a functioning gene, and as soon as the functioning gene goes into the cell, you will start producing the normal protein, and then whatever symptoms you were experiencing should now go away. Okay. Now, typically, for this gene therapy to work, the condition you're trying to treat has to be a recessive condition. And the reason is because if the condition is a dominant mutation, it doesn't matter how many normal copies of the genes you supply, the dominant mutation, by definition, is going to override all of these functional genes. Okay, that's what dominant means. Dominant alleles means you only need one copy of it for it to be manifest, right? For it to show up in the person. So like replacing replacing the gene with a normal copy gene is not going to fix the problem in this case. To fix it, you probably need a different approach. You need something that would override the dominant gene. Okay, but we're going to get to that later. So an example of that would be sickle cell anemia. This is caused by a missense mutation. Once you, again, supply the normal hemoglobin gene, you will start to produce normal hemoglobins, and then you will be able to transport oxygen in the body with no problem. Beta thalassemia is another type of anemic condition. This one is caused by a nonsense mutation. Right, so the hemoglobin is missing a chunk of amino acids in a structure, and it's not able to deliver oxygen. There are some minor cases, but then there's also like severe cases. It's called thalassemia major and minor. And in severe cases, people need to constantly receive blood transfusion just to survive. Okay. And this constant transfusion actually caused them to have iron overload. And then so they have to treat that as well. Right. So it's something that you want to fix. And because it's a nonsense mutation, it's an ideal candidate for gene therapy. Once you supply the normal copy, you should be able to make the normal hemoglobin. Hemophilia is something I told you about. This is an X-linked recessive condition with a bleeding disorder. Okay, you cannot produce blood clot factors, and you bleed out for a substantial amount of time. And then, if you provide the recipe for the normal clotting factor, then you're okay. Okay, you gotta understand what I'm saying. I'm not saying to supply a whole chromosome. Okay, it's not like, oh, these, the X chromosome's not working. Let's give them like a complete X chromosome. Right, that's not what I'm saying. We're only replacing the missing gene. Yeah, we want to insert the gene into the X chromosome. Ideally, the best case scenario is to cut out the old one and then literally replace it with the functional one. But that requires a lot of precise control. there is some new technology called CRISPR. You might have heard of it that allows very precise cutting and editing. So we're getting there, but I don't think we are 100% there yet. By the way, there had been successful cases of using gene therapy to treat these conditions. Okay, so this is not science fiction. We have the ability to do it, but there is still a lot of room for fine-tuning the technology so that it works better and it's going to be able to apply to more situations rather than just these limited diseases. Okay, next one is suppressing a gene. That's the second goal. First goal is to replace the defective gene, but as I told you, it's not always possible. Okay? Sometimes you have a dominant situation, then you cannot replace it with a functional gene. And then the next best thing would be to suppress the overly active gene. We already talked about oncogene before. A proto-oncogene is a normal gene that promotes cell division, but oncogene is a mutated version of that. It's a green light that doesn't turn off, right? And so you have cells that are undergoing cell division very rapidly, which could cause tumors, right? So in this case, if we use gene therapy to deliver a blocking gene, the blocking gene is going to basically shut down the overly active oncogene, and then the cell, in theory, would return to normal function. So, you have a green light that's always on. Think about like an actual intersection. If the green light doesn't shut down, then what do you do? Well, you can take some kind of covering and then put it over the traffic light, right? Then we don't see it anymore, right? Um, the light is still kind of there, but we are blocking it in a way. And that's what this gene therapy is. With the blocking gene, we cover these, we suppress these overly active genes. The last goal is to modify the clinical course of a condition. Right? So in this case, uh, take cancer, for example. Researchers is trying to insert what's called a suicide gene that would trigger cell death. Okay, so these suicide genes get delivered to diseased cells, and then once it goes into the cancer cell, it would produce some kind of toxic product that would trigger apoptosis. Right? It would kill them off. Another way to do this is to insert a marker gene. One of the challenges of treating cancer is because unlike bacteria and viruses, the bacteria and viruses, they are from outside of the body. So your immune system is pretty good at distinguish what belongs to the body and what does not. Okay. And when they see something that doesn't belong to the body, they will attack it and destroy it. Right. But cancer arises from your own bodily cells. And so they look very similar to your own healthy cells. And that makes it difficult for your immune system to detect them. Okay. And so the idea of a marker gene is to deliver the marker gene into the cancer cell, and that will cause the cancer cells to produce these special markers on the surface. And these special markers are not found anywhere else in the body. So your immune cells would see these special markers. They're like, hey, that's not supposed to be there. We've never seen that before in healthy cells. This thing must be bad for the body. Let's attack it. Right? So by delivering the marker gene, you basically highlight the cancer cells, and the immune system will do the rest of the work. Okay? Whereas a suicide gene, it's not about the immune system. It's just delivering the suicide gene into the target cell, and then the target cell will basically kill itself because of the products from the suicide gene. Okay, so by killing off these disease cells, you improve the chance of survival, right? You alter the clinical course of the condition. Now, there are two ways to do it, an ex vivo and in vivo. Ex vivo is to take the cells out of the body, change it in a petri dish, and then reintroduce it back into the body. Okay. Ex vivo means outside of the body. Vivo is the body. Okay. So this one, you have more control over the cells because you are manipulating them in a petri dish in the lab outside of the body. You are only modifying these cells, and when you put it back in, you know these are the cells that you've worked on, you've checked to see that they are functioning properly after, the manipulation and before you introduce them back into the body. But of course, that involves more like handling, and sometimes that comes with cost and time as well. For in vivo, you use some kind of vector to deliver the DNA. Okay, so you use a virus. You take away all the harmful parts of the virus that might cause disease, and you replace it with the gene that you want to deliver, and then you infect the person with the virus. Then the virus should, in theory, deliver the gene for you, right? So that's in vivo. And the main challenges of gene therapy is targeted delivery. Okay, how do you ensure the gene is going to go where you want it to go? Okay, sure, the virus can deliver it for you, but and we learned virus injects the DNA into the host, right? But how do you know, how do you control where it is going to be injected? You know, like the suicide gene, it would be very, very bad if the suicide gene went into the cancer cells as well as the healthy cells. So now you're killing off the healthy ones as well. So that's a big no-no, right? Same thing for the marker genes, right? So like, how do you control where it goes? It used to be a really, really big challenge, but like I said, there's something called CRISPR-Cas9. You can look it up, CRISPR, CRISPRE. That is a newly discovered technology. They learn it from bacteria actually, and now it's you can use it to have really precise control as to what to cut out and what to replace. Okay. I think in the next decade or so, based on this technology, they're going to be able to really refine gene therapy, and it's going to become common. And we'll be able to treat diseases that have no cures right now. Immune responses, right? You are introducing foreign material, sometimes using viruses, so you're not sure how your immune system will handle it, right? It's possible that they would become hyperactive in some cases. I've read recently read a book, and in the book it was talking about how a person has a type of skin cancer, and as a treatment, they tried gene therapy, and it was working. The gene therapy was causing the skin cancer to regress, but it messed up the person's immune system, and his immune system started attacking his liver. Okay, so he has very damaged liver, and so they had to stop the therapy, gene therapy, and so the his body stopped attacking the liver, but then the skin cancer came back, right? So like there's this delicate balance of, how do you attack uh, only the cells that you want without triggering alarm for the rest of the body, right? And that's, a key thing that they have to overcome. Interaction with other genes. Again, this comes with a target delivery aspect. If I insert a gene, what if the gene goes into the middle of an other gene? That's gonna destroy it, right? So we have these off-target effects. And we talk about, putting a cover on the green lights, right? But what if you put a cover on not just the oncogene and other green lights are affected as well, right? Like you're not really sure how the genes will interact with everything else that's going on in the body. Sometimes what happens in the lab is not necessarily going to be what's happening in the body, right? And you really don't know until you actually go ahead and do the treatment. Cost is very, very expensive, especially for individualized treatments. A lot of these treatments are not one size fit all. They have to really tailor it to the person. We're talking about, $500,000 to $1 million per treatment easily. And sometimes insurance don't pay for it. Right now, it's not bad because the researchers they actually invest all these money into these technologies, and they want to figure out a better way of doing it. And so sometimes they pay for the treatments. But then, as this becomes more and more popular, people might have to pay out of pocket for it. So there's an aspect of access, right? Is everybody going to be able to access these kind of therapy in an equitable way, right? There are factors, socioeconomical factors, all these things that needs to be taken into consideration. And that is it for lecture 9. Okay. Please remember the quiz is this Wednesday, and you will have to solve problems, the monohybrid, dihybrid stuff. So make sure you are prepared for them. Now, I do want to move on to lecture 10. Uh, it's a it's a nice lecture that helps you bridge the transition from biology to pre-anatomy and physiology, which is what you'll be learning next semester. Okay, the physiology and anatomy, it talks about like body parts and how it works. It's 6 hours per week. You'll be learning about a body system every week, starting with the cardiovascular system. And so, we understand a lot of the biological processes that's happening now from this course, but how does this translate to like the bigger organisms, right? That's basically the focus of the next semester. Okay. So let's start with lecture 10 here. Okay. This is called body tissue. Okay? Now, when we were first created, okay, when the sperm meets the egg, we were single cell, zygote, okay, and then it divides. But I remember someone asked, maybe it was this class, maybe it was another class, they were asking me, how does that single cell become all the different cell types in the body? How does it become like the brain cells, the heart cells, and so on and so forth? So this process of becoming a particular cell type in the body is called differentiation. Okay, differentiation. And when a cell differentiate, they become specialized. Okay? And differentiation often is a one-way transformation. Once they become a brain cell, they stay as a brain cell. They cannot go back and then choose to become a heart cell later on. Okay? And at the heart of it, the all the cells that create all the cells in the body, all the different cell types in the body, ultimately came from stem cells. Right? So stem cells are cells that can specialize and differentiate into different kinds of cells. These are the cells that can become the brain cells under the right condition. They could become the liver cells. They could become the skin cell under the right condition. Right? And as an embryo, when we're still growing in our mother's womb, we had a lot of embryonic stem cells. So these embryonic stem cells are very versatile. They literally can become any cell type of the body. But once the person is born, then we have less versatile stem cells and even less when we become adults. Okay, so these adult stem cells, they are more restricted within one category of cells. For example, you have stem cells in your bone marrow. Inside your bones, you have bone marrow, and in the bone marrow, you have bone marrow stem cells. Those stem cells are restricted to becoming blood cells. So they could become red blood cells. They could become different kinds of white blood cells, right? So it's more restricted to a category. They cannot become brain cells, for example. Right? Whereas embryonic stem cells, they can become anything. Right? So as adults, we have limited pools of these stem cells. So stem cells are self-renewal as well. Okay, so self-renewal. That means they can make more copies of themselves. Okay. So I don't know if there are any mothers here, but if you gave birth in Canada, one of the options you were maybe given was to store your baby's cord blood. Okay, so the umbilical cord that connects you and the baby, they could actually extract the stem cell from the cord blood, the baby stem cells, and you can freeze them down, okay, for a fee, of course. And the idea is, if you freeze down the stem cells later on in life, if your child becomes sick with certain kind of diseases, they could use the stem cell to treat them, okay, in cases like leukemia, for example. Okay, but It's quite expensive. I mean, I didn't do it for my kids, but I know people who can afford it. That would be an option for them. Okay. Now we went through quite a journey in this course. Okay, we started with atoms and molecules at the beginning, and then the macromolecules in lecture two. That seems like a lifetime ago. And then we talk about the organelles and a bunch of things that's happening in the cell, right? With the transcription, translation, DNA replication, and all that stuff. And now we're basically learning tissue in this lecture. And like I said, next semester you'll be learning about organs and organ systems. These are all the body system that we'll be learning about again next semester. Digestive system, respiratory system. It's heavy, guys. I am gonna warn you. I teach that course too, but I don't think I'll be teaching you guys next semester. Someone else will. But it's a very heavy course. Yeah, it's much more difficult than this one because there's a lot more details you have to remember and yeah, yeah, I'm sure you guys will adapt to it just fine. Okay, what we're gonna do now is we're gonna learn about the body cavities and what's inside them. I mean, a lot of it is common sense. Okay, so let's just go through a few things here. Up here in the head, you have the cranial cavity. Cranial cavity. What is in here? I'm sure you know it's the brain. That's where the brain is. And don't worry about the vertebral canal. That's where the spinal cord is. Again, don't worry about it. Here is the thoracic cavity right here. All right, thoracic cavity. Can you guys just tell me some things that in it? You can list it out and you tell me what do you find in the thoracic cavity? Okay, we have lungs right here and hearts. Good. What else?
Trachea,
Trachea. Excellent. That's the airway for anyone who does not know. Yeah, there are various blood vessels as well, but these are some key ones, okay? Now you have a muscular muscle here. It's called a diaphragm. It's like this. It's right here. It helps you breathe, okay? It moves up and down, and then it pulls the lungs down and push the lungs up, and it helps you breathe in and breathe out. And the diaphragm separates the thoracic cavity from the abdominal cavity. Okay, there are a bunch of stuff here. Again, just hit me with it. Okay, what do you know? What's in there?
small intestine, large intestine, liver,
Oh, a bunch of stuff. Oh,
spleen,
yeah, okay. Thank you. Small, large. Anyone else? Intestine. Okay, spleen. That's a obscure one. If you don't know what the spleen is behind your stomach. On the left, okay, it stores blood for you and it does a little bit of immune function for you.
pancreas,
Sure.
pancreas,
Yeah. Okay, so like just a few, right? Just a few of the things that people are naming. I on the test, I'm not gonna give you obscure stuff, okay? Like people might not have heard of pancreas, so like I'm probably not gonna put it there, okay? But like large intestine, small intestine, stomach, that's all in the abdominal cavity, okay? Liver is another one, okay? Pelvic cavity, what's in there? What's in the pelvic cavity? Yep. We have the uterus, good. Although this is a male, but that's okay. This is female only. And the bladder, urinary bladder, yeah. Anything else? There's no uterus in men. What do they have instead? Starts with a P. Do we have any males? Oh, thank you. Okay. I was
prostate
gonna
gland,
prostate gland, yes. Prostate. Okay. So these are all in the pelvic cavity. Again, don't freak out. Okay, like you don't have to memorize all of these things. It's just I'm just showing you what's in there. Okay, and if I'm gonna ask you a question on the test about it, it's gonna be obvious things. Okay, like you would know the stomach is not in the thoracic cavity, right? Stomach is in the abdominal cavity. Okay, but you'll be learning all these next semester. This is just like an overview. Okay. moving on. Anatomical terms. Okay. When we use when we describe the position of body parts, we want to be using anatomical terms. So everybody's using the same set of description, and we're all referring to the person in a particular position. So we're not going to be confused. Okay. What do I mean by that? Well, for example, if I say that your kidneys are below the stomach. Okay, the kidneys are below the stomach. Okay, uh, but you're like, is that true for everybody? Like, well, it might not be true if you're lying down, right? Or if you are sitting. Like, is it gonna matter if you're sitting down or like if you're bending over? Like, what position are you talking about, right? So to avoid those kind of confusion, we will be using a set of anatomical terms, and these terms apply to a person when they are in what's called the anatomical position. Okay, I'm gonna try to show you. Uh, my camera doesn't go very far back, but you can see. Okay, basically, like if you're standing here with your two hands facing forward like this, okay, my toes are pointing forward. This is called the anatomical position. And so all the terms I'm going to teach you right now assume the person is in that anatomical position. Right? So I want you to draw this out, big diagram, so that we can label it. Okay, it doesn't have to be nice. Okay, so there is the person with the fingers and whatnot. Okay, and then you can draw the other hand. Again, it doesn't have to be super nice. It just has to look like a person. Okay, just have to look like a person. There we go. Okay, so this is called the anatomical position. Anatomical position. I know it's a little bit disproportionate, but maybe you can do better than me. All right. Now, once you have it, I'm going to teach you a bunch of terms. Okay, these terms comes in pairs, and they mean opposite things. So let's start with the first pair of terms. Things that are on top, higher up, we say superior. Superior. And things that are further down, we call them inferior. Okay, superior, inferior. Your head is superior to your body. Okay, on top of superior and inferior. Okay, can I move on, or you guys need a moment? Okay, no news is good news. Can we keep going? These are from the PowerPoints. Okay, so we have some terms here. I'll give you a minute to fill it in, and then we're going to take it up. Okay, we are only using the terms superior and inferior. Okay, just fill it all in. Okay, 4 questions. If you don't understand, please ask. Okay, let's take them up now. The hands are blank to the feet. Well, your hands are right here and your feet are right here. All right, so hands are superior to the feet. I'm just gonna put S here, superior. What about the knees and the waist? Knees are here, waist is here. So the knees are going to be below the waist. All right, so that's inferior. Again, when we are talking about the anatomical position, right? It's not like when you're lying down, it's not like, you cross your legs or something like that. What about elbow and wrists? Elbow is here, wrist is here, but again, not like have my hand up, but in the anatomical position. In the anatomical position, the elbow is superior to the wrist. And the calf muscle and the ankle. Calf is here at the back, and the ankle is here. So calf is superior to your ankle. Are we okay? Do I need to elaborate before I teach you like 10 other terms? We do have 10 other terms coming, okay? I just want you to understand superior and inferior for now. Are we good? All right, tough crowd. Let's keep going. The next set of rules we have would be, let me see what's in the PowerPoint. Okay, anterior and posterior. Okay, so we can look at the person from the side here, and we might have to draw another one here. Okay, this is from the side. The person is standing. Okay, and then the hands just like before. All right, so over here, another pair of terms. All right, we have things that are at the front and things that are at the back. So at the front, we say anterior. And at the back, we say posterior. Now, sometimes anterior is also called ventral, and then posterior is called dorsal. Dorsal. Okay. Anterior, ventral, posterior is dorsal. Now, uh, in humans, it's the same. Okay. Ventral actually means the tummy, okay, where the abdomen is. Dorsal is like the back, but it's not always the same for every organism, okay? Like if you think about a dog, for example, okay, like a dog or whatever, dog, okay. Then in this case, this is anterior, this is posterior, and now this is dorsal, and this is ventral. Okay, so dorsal and posterior are the same for us, and anterior, ventral are the same for us because we walk on two legs. But for things that walk on four legs, then these words kind of mean um, different things. Okay, in like shark movies, right? You always see the shark fin coming, right? Yeah. Do you know what this shark fin is called? Which fin is that? Want to take a wild guess? That would be the dorsal fin. Okay, dorsal fin, the back fin of a fish. Okay. My point is, even though it's the same for us, it's not always the same in other contexts. Okay. Not important. You don't have to write this down. This you should know though. Okay. Let's take a look at another set of questions. There we go. I'll give you a moment. Now, it's possible sometimes you can use more than one correct answers. But here we are asking for anterior and posterior only. Okay, so please Fill that in, and we're gonna go from there. If you don't know where these things are, um, I'll tell you later. Okay, so heel is here. That's the heel. That's the toes. So heels are posterior to your toes. Bicep is here. Tricep is here. Again, you're in anatomical position. So the biceps are in front of the triceps. Right? We say anterior. Okay, if you think about your legs like this, just like you're sitting there right now on the chair, this is not in an anatomical position, but I'm trying to tell you. This here, your thigh, that is the quadriceps. Quadriceps. And then here, it's the hamstring. Right? So now, same thing, but standing up. The quadriceps will be anterior to, oh, sorry, the hamstring. The hamstring will be posterior to your quadriceps. Right? That's why I said it's important to have a standardized position for everybody. Because if you're sitting down, then the quadricep is actually superior to your hamstring. Okay, but no, we are always talking about the anatomical position, the person standing up with the hands apart. All right, there we go. Again, on the test, I'm not going to give you like weird body parts that you've never heard of. It's just going to be the stuff we did in class or common stuff. Okay. All right, let me see what's next. I just have to do it in the same order as the PowerPoint, so it's not going to confuse you. Okay, next one is this. If we if we divide the person into left and right with a imaginary line here. Okay, this is called the midline. Okay, so things that are farther away from the midline.
Is it the lateral?
Yes, it is lateral. Thank you. Very good. Okay, so that is lateral. Yeah, if you have taken anatomy before, you would know that this is lateral. And the opposite of lateral is medial. Medial. Your ears are lateral to your nose. Okay? Ears are lateral to the nose. The nose is medial to your eyes. Okay, nothing is more medial than things that are on the midline. So what are some things that are on the midline? You got your nose on the midline. What else is on the midline? The mouth is on the midline. Yeah. Anything else? What's on the midline? No. Yeah. The neck is on the midline, right? The belly button is on the midline, right? So all these things are medial. Nothing is more medial than these structures. Okay, so on the side, yeah, the spine. There you go. Tons of stuff in the midline. Your airway, if you want to go inside, right? The esophagus, for example. Again, nothing crazy, just basic stuff. Okay, don't worry about the esophagus. If you don't know what that is, you'll learn it next semester. Okay, here we go. Medial and lateral. Okay, go, go, go. Okay, so the arms are lateral to the midline. The neck is medial to the arms. Shoulders, shoulders are lateral to the midline. And the ears are lateral to the nose. Okay. Any questions? Anyone not sure why some of these are the way they are? Just let me know and we can talk about it. I hope this is relatively simple so far. Okay. Certainly it should be more simple than the big B, small b stuff. All right. And other pair of terms. This next set is used to describe the limbs. Okay, so either describe the stuff on the arm or on the legs. If it's further away from the joint, then we say distal. And if it's closer to the joint, then we say proximal. Right, so the joint for the arm is the shoulder. If it's closer to the shoulder, then it's proximal. If it's further away from the shoulder, then it's distal. For example, your fingers will be distal to your elbow. Right? Distal to your elbow. Your wrist would be proximal because it's closer to the joint than your fingers. Right? The same thing applies to stuff that is on your legs. Okay, so if it's closer to the hip, then it's proximal. Again, it's all in reference to the connection point. The connection point being the hip for the leg and the shoulder for your arm. Proximal and distal. Okay. Again, another set of questions. Please try it out. All right, fingers are distal to the elbow. They are further away from the shoulder. Uh, Knees are proximal to the feet, and the wrists are proximal to the fingers. Right, now you might be wondering, hey, can I say that the wrists are superior to the fingers? Yeah, that works too. But then like proximal would be a more appropriate term because we tend to use proximal and distal when we describe structures that are on the arm and the legs. Okay? But keep in mind there are more than one possible answers in these situations. Okay. So these are all of it. I can't really draw the other ones. The other ones are hard to depict on a picture. But let's go through the PowerPoint slides and I'll show you. Okay, so next is ipsilateral. Ipsilateral means same side, and contralateral is opposite side. Right, so like the midline is dividing the body into left side and right side. So for example, we can say the spleen. Someone mentioned the spleen earlier. Spleen is here. Gallbladder is here. So the spleen and the gallbladder are on the opposite side of the body. So we say contralateral. Contralateral. This one may not be the best example, but you can see stomach is here, and the liver is quite big, but bulk of the liver is on the right side, and then the stomach is on the left side. So we say this is also contralateral. Contralateral. If you want an example of ipsilateral, you can say gallbladder. And anybody know what that little dangling thing here is? Yeah, that's the appendix. Yeah, so these things would be ipsilateral. All right. Contra is opposite. Ipsi is the same. And then finally, we have deep versus superficial. Deep versus superficial. If something is deep, it's closer to the core of the body. If it's superficial, it's closer to the skin. So bones are generally located something muscle. So bones are inside muscles, right? Not the other way around. Like you would know if you think about eating like chicken wings, right? The meat is outside of the bones. So we would say bones are located deeper than muscles. Skins are superficial to muscles. The brain is blank to the cranium. Cranium is your skull. So your brain should be inside the skull. The pericardium, and even if you don't know what the pericardium is, just keep reading, right? The pericardium is a membrane that surrounds the heart. If it surrounds the heart, it must be outside of the heart, right? That's what surrounds means. So that would be superficial. All right. That is that. Okay. Are we okay with all the anatomical terms?
Yes.
Yeah, thank you. And anyone else have any questions before I tell you the rest of the story? All right. Next, we're going to talk about planes and sections. And basically, there are three planes, okay? Let me try to, I think I have a picture in the study guides. I don't know if you guys print this stuff out. Hopefully you do. And if you do, you would have this. Um, See, there is more pictures here for you to fill in if you want. Here is another one though that I need right here. Let me just copy this person over. There we go. So this person is separated using various planes. So for example, we can have a plane that, let me see, is it working now? Okay, we can have a plane that separates the person into superior and posterior portions. Okay, like this one right here. This is called a transverse plane.
Horizontal. Can you
Yeah,
say that?
horizontal plane, transverse plane. So this separates into basically top and bottom, okay? Superior and inferior. When a person goes for like a CT scan or X-ray scan, then you are viewing the body parts you from different planes, right? So that's a transverse plane. And over here, let's do another one. By the way, you're gonna need a lot of colors for next semester, okay? There's lots of diagrams that you have to color and label. So having colors will make the process a little bit easier, I think. Okay, so this one is called a sagittal plane, and it separates into left and right. Now look at me for a second. This is a sagittal plane. It can cut anywhere, right? It can cut here, here, like here. If it goes down the midline, then it's called mid-sagittal. Otherwise, it's just regular sagittal, okay? And then one more over here. We have A here that divides it into front and back. Front and back. Okay, you can fix up the coloring later. Depends how you want to be looking at it. And this front and back is called the frontal or coronal frontal plane. Okay, and it separates into anterior and posterior. So next semester, when you're labeling a diagram of the heart, for example, or of the brain, you have to know which cut is it. Like, because cutting it from different perspective will review different structures sometimes, and you will have to know how to label them accordingly. Okay, this is the brain, and you can see this. These are from like different sections. Okay, this one is frontal. This one is transverse, and this one is sagittal. So when someone do like a CT scan, like I said, you will be able to look at the body from different planes, and they could actually be constructed to create like a 3D view of the structures. Right? Did you know you have holes in the brain? These are called ventricles, and they are filled with fluids. You will learn that next semester. All right, moving on. Let's do one question here. All right. So choose all the appropriate terms. Okay, so the right thumb is something to the left big toe. Well, certainly it is on top of it. It's above it. And it's also lateral to it. Yeah, remember, your hands are spread out more than your legs in the anatomical position. And one is on the right, one is on the left, so it's contralateral as well. Contralateral. Okay. Um, this one always gets people. You have to think of the fish, okay? Think of the fish, and then this is a cut that's produced when you cut the fish like this. Okay, so because it separates it into front and back, anterior and then posterior, the answer is coronal or frontal. Okay, coronal or frontal. All right. Now, we're not going to be able to finish this lesson today, which is fine. We'll finish up on Wednesday. But I do want to take another 15 minutes or so just to go through a little bit of it. Okay. So the next portion here, we're going to be talking about the different kinds of tissues. Okay. And we're going to start with the epithelial tissue. Basically, what are tissue? Tissues are a collection of cells for a specific function. Okay, so you have different cell type that makes up a particular kind of tissue. You can have connective tissue, you can have epithelial tissue, you can have nervous tissue. So these tissues are made up of not just one cell type, but a bunch of cell types with organic materials around them usually. And we're gonna just do epithelial tissue today and finish the rest later. The general characteristics of epithelial tissue is that it has one free surface. That's the side that's going to be facing the environment. Okay, that's going to be the side that's facing whatever fluid is passing through it, and we call that the apical side. So if you imagine like the intestinal tract here, uh, this is the apical side. That's the side where the food is going to be passing through. That's the apical side. If you think about the airway, if this is the airway and the air is going through this hole, then my finger, what I'm touching right now on the inside, that's the apical side. So it's in contact with the external surface, exposed surface. The other side that's connecting it to the rest of the body, we have what's called the basement membrane. The best way to visualize this is to think of a doormat, okay? The side you are stepping on, that's the apical side. And then if you like flip it and look at the bottom of it, what's stitching all of these things together, like this gray beige looking structure here, that would be like the basement membrane. Okay, so the exposed side is the apical side, and then the underlying side is the basement membrane. So all epithelial tissue has this kind of structure. And we can, instead of saying epithelial tissue, sometimes they just call epithelium. It means the same thing. We can classify epithelial tissue based on number of layers and their shape. Number of layers and their shape. If it's a single layer, then we say simple. If it's multiple layers, then we say stratified. Okay. One layer, simple. Two or more layers, stratified. If it's a flat cell, like an egg, sunny side up egg, with the nucleus in the middle, like that, that's called squamous. If it's a cube shape, like a cork box with the nucleus there, we say cuboidal. Cuboidal. Okay, think of this as a Y, cuboidal. If it's rectangular in shape, like a fridge with the nucleus there, then that's a columnar epithelial. And then we can use these words in conjunction. If I say simple squamous, then it's one layer of these flat cells. If I say stratified squamous, then it's multiple layers of these flat cells. Okay? We describe them by number of layers and their shape. Now, this is going to be overwhelming, okay? I am not going to lie to you, but you are not supposed to memorize all of these things, okay? You just have to understand the overall key concepts. Okay, I don't expect you to memorize exactly where they're from and then, all these functions. I will try to boil it down for you. Okay, so let's start with simple squamous. What does it mean? Simple means one layer. Squamous mean flat cells. Okay. So where do you want to have a single layer of flat cells? Well, if it's just a single layer of cell, it's really easy for things to pass through. And so it's perfect for things like diffusion and osmosis. Okay, never mind filtration, don't worry about it, okay? So where do we want diffusion to occur? At the lungs, at the lungs. So air sacs of the lungs. Okay? When we breathe in oxygen, we want it to be able to cross the tissue in the lungs and go into the bloodstream. Right? So we want simple squamous there. You don't want multiple layers. It's really hard to pass through. One layer is good. Okay? Other places, blood vessels and heart linings. Again, don't worry about them. It's good to remember if you can, but it's not detrimental if you don't. Okay? Just like lungs would be good. For diffusion. Next, we have stratified squamous. What does stratify mean? Stratify means multiple layers of flat cells. Okay, so individually, maybe they're not very strong, but once you stack them together, if you have many, many, many layers, then they become quite strong. And in fact, you want to have stratified squamous at places you need protection for. Right? There are two different kinds. You don't need to know the two different kinds. I'm just telling you. One kind is keratinized, which makes it waterproof. So that would be useful for like surface of the skin. And then you have non-keratinized ones, which are not waterproof. Right? So where do you need protection? Well, you need protection for the palm of the hand with grabbing things every day. There's a lot of rubbing, frictions, wear and tear. Bottom of your feet, right? In fact, if you wear sandals a lot in the summer, your skin at the bottom of the feet might get a little bit thicker, right? That's the stratified squamous layer, getting more layers to protect you. Okay. Sometimes you use the hands, certain tools too much, you start developing these calluses, right? That's basically stratified squamous, adding a few more layers. Okay? Your body wants to add more protection to places where it's lacking. Okay. Now, inside the body, inside the mouth, for example, inside the esophagus, which is like, your food tube, lots of things go through it every day. Hot stuff, cold stuff, acidic stuff, right? Spicy stuff, right? Sometimes you eat things that are hard, like chips. All these things goes down and it's rubbing against the esophagus, and we need some protection from these kind of abrasion. Okay, and that is why we have these stratified squamous. Okay, so simple squamous, no protection, so thin. Okay, and things just go right through, and that's okay. We need it for diffusion. Stratified squamous, not good for things to pass through, but so many layers, it offers more protection. Okay, that's about as much as you need to know. Okay. And I mean, like, think about where do you need protection? Okay. Do you need protections on the surface of your eyeball? Okay, like for example, this is a legitimate question. Do you want simple squamous on the surface of the eyeball, or do you want stratified squamous on the surface of the eyeball? What's more appropriate? Anybody? Think of the eyeball here. Okay. Do you want like 10,000 layers here? Or do you want, just a thin layer? What makes more sense? Do you need a lot of protections on the surface of the eyeball? What needs to go through your eyes for you to see? Light, right? Okay, you need light to go through, right? So then, you want the light to be able to pass through the eye with minimal impediment, right? You don't want to have many layers here. If you have too many layers, then the light is not going to be able to pass through easily, right? So for the surface of the eyeball, you would want simple squamous as opposed to stratified squamous. Is this okay? I hope so. All right. If you don't understand, ask. All right, moving on. Next, we have simple cuboidal. Simple cuboidal. Simple is one layer. Cuboidal is square cube looking. Okay, so these are they gonna offer a little bit of protection, but the main function is they forms the lining of a duct. Okay, so again, my tissue paper roll. This is a duct, and inside the duct there could be various things. Okay, it could be sweat, it could be milk, it could be urine, depending on where you find the duct, right? So this is the inside, that's the apical side, and all these struck stuff right here, the actual tissue paper that I'm showing you, that's where the cells would be. That's where the cuboidal cells are gonna be, and it looks like this, right? So that's like a one cuboidal cell here, and then another cuboidal cell, and then it's just a single layer that builds the entire tube, and they are good for secretion and absorption. Okay, they would be secreting things for glands, and they would be absorbing things. Um, I mean, it's difficult to give you precise examples without going into much details, but like in your kidneys, for example, you have these cuboidal cells that would help absorb minerals, salts, that kind of stuff back from your urine, in case you don't want to get rid of it from the body. And also in other places in the intestinal tracts, you might absorb nutrients as well, right? So these are good for secretion and absorption, single layer. And then here we have simple columna, which are like the bigger ones, okay, and also one layers. They are also for secretion absorption, but it offers more protection since they are bigger. And so again, you will find them in the in the GI tracts as well as in the ducts in some cases. Okay, so there are no stratified cuboidal or stratified columna. Only single layer exists for cuboidal and for columna. And I'm just going to show you two more types. Over here, it's called pseudo-stratified. Pseudo-stratified. Pseudo means fake. Okay. Ciliated means they have little hairs on them. Those are cilia. We learned that before. So these are hairy columnas that looks like they are multiple layers, but in fact they're not. Okay. It's fake, pretend to be multiple layer. That's what it means. Okay. So pseudo stratify, it's single layer, but it appears to be multiple layers. And you will find them in airways. You will find them in reproductive tracts of the female, okay, to move the eggs. That's the pseudo stratify. And some of them, instead of having cilia, they have these foldings. Okay, so these foldings are part of the cell membrane, whereas the cilia, they are more like brushes, like different structures. Okay, so cilia like this, these are called microvilli. Again, don't worry about the names. Okay, it's not like I'm just showing you different kinds, and they are there to help absorption of nutrients. Okay, so I will summarize this for you next class, but first, maybe we can try this out. Okay. Let's start with this one. This one might be easy. Okay. What do you think this is? Okay. Is it squamous? Is it, you know, cuboidal, columnar? What do you think this is? Right? This is like a side view, if it helps. Okay, this is looking at it from the side. Okay, you can see that they are kind of square in shape. Okay, so these are actually cuboidal. Great for absorption and secretion. Cuboidal. And you can see this is actually the duct. These are actual pictures, by the way. That's the duct, and that's where you're gonna have like various secretions passing through. This is a duct. Okay, maybe this one. How about this one? If this is cuboidal, which one would this be? Okay. These are more rectangular in shape. So those are columna. Okay, you will have to identify these pictures, okay, um, on the test. But again, nothing crazy. It's just what I'm showing you here. Okay, this is also simple, one layer. Okay. How about this? Okay, look, this is like so many layers, so many layers.
Stratified squamous.
Yeah, this is stratified squamous, which again is good for protection against wear and tear. You will find it at the bottom of your feet, the palm of your hand, inside of your mouth, okay? Esophagus, that kind of stuff. These one here, it looks like it's multiple layers, and you can even see the cilia, the hair. So this is pseudo.
Stratified.
That's right. That's the one with the long name, okay? Pseudo stratify, pretend to be stratified, and then ciliated columna in the airways and stuff like that. Airways, reproductive tracts. And that leaves us with this one. This is a top view. Okay. I guess that's why it's a little bit difficult to see. This is top view as opposed to all these other ones are side views. This is simple. Simple squamous. Okay. Simple squamous for just diffusion. Okay. No need for too many layers. Okay, so I'm just gonna go through a few questions with you, and then we'll be done. Okay. Based on everything I told you, are you able to answer these? If you are, then you should be okay for this portion of the test. Which of the following best describes multiple layers of, I'm just relaunching the poll, of multiple layers of flat cells? And just choose the right answer. Okay, participate if you can. Multiple layers of flat cells. Oops. Okay, good. Many of you got it right. It is stratified squamous. Great for protection against wear and tear. Okay. How about this? Best for diffusion, for things to pass through without obstruction, without too much resistance, just goes right through because it's so paper thin. Yeah. Anyone else? It's good. Okay, simple squamous, right? So like I said, big ideas. That's what I want you to remember for this portion of the lecture. Okay, not the fine details. Okay, so that's good for diffusion, right? Okay, how about these things? What are they good for? What's simple cuboidal good for? That's good for secretion and absorption. Some kind of duct, right? Okay. Do we even have like simple columnar? That's another one for absorption and secretion. There is no such thing as stratified columnar. We talked about that already. There's pseudo stratify, fake ones, pretending to be stratified, but there are no stratified ones. Yeah, so this would be like something that requires a little bit of thinking, but just think along the same line we were talking about. Okay, correct answer is stratify squamous. These are the stuff that offers you the most protection, right, against all the stuff in the smoke that the person inhaled, right? Remember, stratified columnar doesn't exist, okay? And pseudo stratified columnar, that's basically the ciliated columnar, right? It's the same stuff. So we wouldn't be replacing it with the same thing, okay? And the correct answer is B. So that's where we're going to leave things at for today. Next class, we only probably need like 1 hour to finish the lecture, and then with the second half, I will take any questions you might have for the final test. We're not gonna have a class next Monday. You can just use it to study for your final test. I'll post like a review package for you to go through it. But let me just pull up the calendar real quickly here, just to go through what's going on here. So you guys are here, and yeah, we were supposed to have the quiz today, but that has been moved. So you'll do it on Wednesday. We'll have a class on Wednesday, and then next Monday you just review on your own. Um, of course, if you have a question, you can email me. And then next Wednesday will be the final test, which involves 6 to 10 rather, plus lab number 2. Okay, so this Wednesday would be our last face-to-face class, and then next week will be the final. Okay, thank you. See you guys on Wednesday. Bye for now.